welcome everyone. Today we have a real special guest. This podcast is for the all hands of Docker, December 2022. And today we have Chris Miller from Washington. And why is he here? Because he's in the WashU, which is Washington University School of Medicine, and which is basically a team of bioinformatics developers, and they use Docker for their research for analyzing the genome sequence and data cancer of patients, and they also do many collaborations with other institutes, right? And as in the words of WashU's Jason Walker, this really helps to accelerate science and medicine globally. So this will be a very interesting and exciting episode. So welcome, Dr. Chris Miller. Please introduce yourself. Thank you so much.
Hi, it's great to be here. Yeah, I'm Chris Miller. I'm an associate professor here at Washington University, and I study cancer genomics and bioinformatics.
Nice. So I would like to start with the introduction. So what is bioinformatics? How can we define it?
So simply put, yeah, sure. Simply put, bioinformatics is just applying computer science techniques to biological questions. And in our case, we study human disease with a focus mostly on cancer. So by sequencing the DNA of people with these diseases, we can try to sift through and find the mutations that are in this people's DNA that cause their specific cancer. Um, Increasingly now we're able to also pair those up with therapies that target their specific flavor of cancer. And so there's lots of exciting research around that, ranging from better detection of cancer early on, through things like simple blood draws to doing things like improved treatments, immunotherapies or cancer vaccines that weren't available, years ago.
Nice. So this is very useful and great. So first of all, thank you for what you do. And I'd like to ask a bit more about you, if that's okay for you. So I'd like to know more about you yourself. How did you start working in bioinformatics? Because it looks really challenging, not easy at all. Basically, it's a mix of different technologies and skills. So informatics and other many more medicine. So how has been? Can you tell us more about your experience and your career? Because it's very interesting. Let's see. Thank you.
Sure, I started out as a biology major and started doing a little bit of computer science here and there. Ended up double majoring in undergraduate, and then went to graduate school for my PhD. And then I've been working on cancer genomics here at WashU for over a decade postgraduate school. Um, that said, a PhD isn't necessary to get involved in this field. We've got a whole spectrum of people ranging from, hardcore programmers who optimize algorithms and such, and to people who are bench scientists who just, learned a little bit of code to be able to analyze their data. And really makes our team works well is that we have like that expertise across the whole spectrum of it, and we're able to bring those to bear on the problems that are important, like cancer.
Nice. Thank you. And now I'd like to ask you about some numbers, because I don't have an idea on these numbers. So I don't know how many patients are involved in this research. So can you give us more like numbers to have an idea of the size of this whole research institutes? Thank you.
Sure. I mean, that really depends on the study. I mean, we might be doing big projects where we analyze the genomes of tens of thousands of patients to try to understand general trends about how cancer forms. Doing that on the cloud and doing things at scale. And other times we're working on single patients, doing a workup of one person's genome to try to inform their treatment in real time, to understand why their cancer relapsed, and whether we can have a better drug to treat it. And so that's the idea of personalized medicine. And we're trying to really make that a reality for every patient.
Nice, nice. Thank you. And in the introduction, I talked about other institute in which you collaborate with. So can you give us an idea which are those institutes that you collaborate with, and how do you collaborate with them? Thank you.
Sure. I mean, all of science is kind of fundamentally collaborative, and we benefit from being part of a huge community of universities across the not only the nation but the world, working on similar goals. people designing algorithms and sharing them, and just contributing to that large pool of knowledge we need to figure out how cancer works. And that ranges from giant centers like the Broad Institute in Boston that sequences tens of or hundreds of thousands of genomes to, small labs with only a couple of members that carve out a niche studying one particular gene or one small aspect of a disease. So all of them have a role to play, and by sharing our code and making it available, we're all working together toward a common goal.
Nice. Thank you so much, Dr. Miller. Okay, so now I would like to go a little bit more into the main topic of this interview. Why are you here? Because there is this event by Docker. So we would like to know if you can give more details on how Docker can help you in this daily work. It can sometimes can be very stressful, very overloading. So which can you go, if you can, a bit more into details in how Docker is helping you, if it's helping you?
I mean, sure, there's a few different ways that Docker helps us. First of all, the workflows we use to analyze these cancer genomes aren't just straightforward single scripts. They're complex pipelines that rely on dozens of different tools. We have to line data to the human reference genome and sift through it. Where in those 3.2 billion base pairs is there a mutation that might be causing the cancer? And then we have to filter them and categorize them using different knowledge bases so we understand which ones are important in driving the cancer. And so this results in kind of big, hairy pipelines that rely on lots of different code repositories, developed in lots of different languages by different groups. And so Docker allows us to kind of modularize that, build complex pipelines that chain these all together with a minimum of having to wrangle installation and dependencies, and also allows them to be run on all the different architectures we use, which range from those big projects happening on the cloud to analyses I'm doing on my own laptop. Um, I would also say that the field is evolving really rapidly. It's often the case that we want to swap in a brand new algorithm for one that we're already using, and having that kind of modular Dockerized workflows really allow us to make that happen rapidly, as opposed to having to rebuild things from scratch. And I think the final point I would say is that reproducibility is just absolutely essential in science. In order to trust the results from another group, I need to be able to replicate those. And so by locking down the environment and allowing others to reproduce that exactly, Docker helps us make those results reproducible to the rest of the community.
Perfect. These words are really warming my Docker Captain heart. So it's very interesting and important to try to have, let's say, other kind of problems, maybe, and not also software problems which can add up on the existing problems. Nice, thank you. And now I would like to ask you a last question about maybe it's more focused for the developers, but I saw that there is a GitHub organization or account which is called Genome/analysis dash workflows, right? There is this repository that you use. So can you
Yes.
tell us more? Can you tell us more about this repository? I see there is an MIT license. Can developers contribute to it so we can give a shout out to this project? Thank you.
Yeah, we make all of our core pipelines and code open source. We're big believers that fundamentally the things that we're working on, this medical research, is too important to live behind firewalls and be locked down. And we want as many smart people working on these problems as we can find. So we absolutely welcome contributions from the community, whether those are from seasoned bioinformatics developers or people just dipping their toes into it for the first time.
Nice. Thank you so much. So this is also why Docker is sponsoring this program. So we're happy for this. I hope that we can help and we can find more people collaborating on this project. We will leave a link in the description. And yes, so thank you so much, Dr. Miller, for your interview. It has been really a great way for me to start the week. And yes, so thank you. And anything else that you would like to add for this, and then we can wrap up. Thank you so much for staying with us so far.
Yeah, no, just thanks for the folks who build the infrastructure and tools that we rely on. You're really making a difference to human health and disease, and that matters.
Nice, nice. Thank you so much, of course, from the bottom of my heart for what you're doing daily. And I hope that we can collaborate more in the future, trying to give support to these great initiatives and projects. So thank you so much, Dr. Miller. And yeah, have a nice day. Have a nice week. I don't know what's the time there. Here is 4 p.m. I'm almost at the end of my day. Probably you are about at the beginning. But yes, nice. So
Yeah, it's 9 a.m. here, so we're just getting started.
yeah, nice, nice. Okay, okay, perfect. Okay, so thank you so much. Have a nice day, and yeah, thank you so much, and see you next time. Okay, perfect. Bye
All right,
everyone.
take care.
Bye everyone. Thank you so much for staying with us. If you have any question, just leave a comment below, and we will reply. But we will leave the links to everything we talked about in the description, so no worries. Bye everyone. Bye bye.